Good evening. Welcome. Who knew? Who knew Microbiomes would draw such a huge crowd? You got, and a rowdy one. Okay. You guys were like lined up out into the parking lot waiting to get beer. I love it. Such enthusiasm for something I have no idea about. No clue. I thought it was like a dome that like people build and like try to make like great, wonderful little small little micro worlds. Way off. Way off. Who's ready? Do what? I'm not that far off? All right, I'll take it. I'll take it from one of the speakers. I'll take that I wasn't too far off. Wait till you read my review after this. I do an article every time. Then you can tell me if I'm like not too far off. Oh, what a fun time. Welcome. Who's never been here before? Okay, all right, not too bad. Who came in December for the first time and came back? Okay, I made you guys sing, right? I made you sing like your favorite Christmas carol. Okay, so we always, we're going to start like a little trend here. We're going to maybe initiate the new people. So if you're here for the first time, stand up. Yeah, it's okay. It won't be too bad. It won't be too bad. So I made the last group sing their favorite Christmas carol. I won't make you do that. I think let's just start off, since it's a new year. How about just at once, just yell out your New Year's resolution. I know it's not going to be too bad. Not going to be too bad. And then you're going to do 25 jumping jacks afterwards. No, just yell out on three. Just yell out your New Year's resolution for just the new people now. Just the new people. All right, 1, 2, 3. Nailed it. All right, you guys can sit down. Mine, of course, to lose weight. My New Year's resolution. Now, you know, the wonderful thing about a diet, the first day is awful, but by the second day, I'm pretty pumped about it because I'm already given up and I'm back off the wagon. So that's my favorite day of a diet is the second day. 2 major announcements this evening and then we'll get rolling. 1, did you know, probably didn't, but did you know that you can submit speakers this year? Somebody read the blog. I love it. So you can go to rtp.org/RTP180, and you can see all of the topics that are coming up for the rest of the year. And then you can submit a speaker if you think that they are worthy to be in that topic. And I'll tell you one that really has my interest, and that is in March. We're going to do the best of RTP180. So if you've been here in the past and you really enjoyed a particular speaker, you can submit that person, and if their schedule allows it, we'll hopefully get them back for March for the best of RTP180. Everybody got it? Second major announcement. This year, we're going to be beefing up sort of the live stream that we do for tonight. So I just was told that 10,000 people are already on the live stream. So if you guys will turn around to the camera and wave to all those people that are watching at home. I inflated that number by like 10,000. But hey, it's the first night, so it'll catch on. I tweeted, if you want to follow me and retweet my tweet, I tweeted the link to the live stream. So you can follow me at Will Hardison. I use the hashtag RTP180. Retweet that because if you're on vacation or you can't make it or the kid's sick or whatever it may be, we're now going to live stream it to where you're going to have amazing video quality. The mics are going to go straight into the camera. It's going to be fantastic. All right, so we'd love to have you guys hop on the live stream if you can't make it to a future RTP180. And with those 2 big announcements, I will go over just a little house cleaning rules, and nice if I turn that on. My name is Will Hardison. For those of you first-timers, I have a small little marketing company here in town. I've been emceeing RTP180 for over 3 years now, ever since we've started doing it. So I'm super excited to be back for another year. You are sitting in what RTP calls the Frontier Building. And it's funny because Michael and I were kind of chatting in the back there, and what a great community that RTP has started to build. And you know, he was saying a couple years ago, you kind of came out to the park to work, you turned around, you went back home. Now there's socials, there's content like this. I mean, there's just so much going on. So the frontier's open during the day for you to kind of come in, co-work out of it, all on this first floor. The chairs are gone, I'm gone. You can get some work done, I promise. But you can come in, co-work during the day, grab a desk, reserve a room. It is completely free on the first floor. And then there's a pretty good waiting list to get some offices on the other floor. So if you're interested, make sure you check it out. Okay, first timers, to let you know, presenters are going to get 5 minutes this evening. We used to have a triangle girl. I got to find, that's going to be me this evening. So I will ding a triangle at the 5-minute mark. That does not mean that they have to finish right then and there. They still get a few seconds to finish up. And then I forgot to tell you all this in the back, but there's going to be time for 2 questions. And again, they're going to be softball questions. I say it just like I say with the clicker, but then with a crowd like as rowdy as tonight, you're going to get a good curveball. If you want to, please get out your smartphone, hashtag RTP180. Please just tweet to us. Use that hashtag. You can follow at the RTP to make sure you're staying up to date there. I told you the clicker gets you sometimes. All right. They are gracious. RTP is gracious to let us use this space, so please pick up after yourselves this evening. If you spill popcorn or your beer, just try to pick it up or let somebody know. We would love to leave this space just like we found it. And then, of course, the biggest rule, and there's no ceiling, there's a cement floor. If you are talking in the back corner, it is going to carry all the way up here. So please just be very respectful to our presenters this evening. Try not to talk during it. If you need to have a conversation, there's plenty of outside rooms that you can go converse or just tweet to each other. That's it. That's all the housekeeping rules. Everybody ready? All right. First up, Julie Horvath is an associate research professor at North Carolina Central University and is jointly appointed as the head of genomics. What? I told them that this is not my normal vocabulary. And microbiology research lab at the North Carolina Museum of Natural Sciences. She is interested in exploring how host and microbial genetics influence human and non-human primate behaviors and disease successibilities. We'll get through it. Just hang in there. All right? Julie, you're up first.
Awesome. Welcome, everyone. So I am tasked with teaching you what microbes are. So let's see how that works. All right. So I want everybody to hold up their favorite hand. Okay. Now look at your hand. What do you see? Do you see microbes? No, because microbes are tiny microscopic organisms. You need a microscope to see them. And it turns out that there are more microbes on your hand than people who live in the city of Raleigh. Just think about that. So when you think about microbes, they include things like bacteria, fungi, archaea, mites, protozoa. And what we used to do is study these in a lab, and we could take our hand and we could touch it to a petri dish in the lab, and we would be transferring the microbes from our hand to that petri dish, and then you could put that dish in an incubator. It's at body temperature, and those microbes grow up and look like a pretty picture, something like what you see here. Doesn't actually work, I've tried it. But what we do now is we have next generation sequencing technologies. And so all the things that live on your hand, there are more things that will live on your hand than you can grow in a lab plate. And so what we do now is we actually take things from your hands. We actually take one of these swabs. That's why I brought this up with me. So we take this swab. It looks like a big Q-tip, and you can just touch it to a surface, transfer it into a tube, and you can extract all the DNA from the microbes that are on your hand or wherever you want. And it turns out that we can then look at the DNA and use that as a fingerprint or a barcode to tell us which organisms we have on our hand or whatever surface we want to look at. Okay? So we're using sequencing to ID the microbes. We're not actually studying them in the lab quite often. And so what this allows us to do is actually look at things from an ecosystem perspective. And we now have what we call these microbiomes. So your microbiome is sort of the ecosystem of all the microbes that live on you. And a lot of the times we study these by using their DNA and studying just their DNA. And so you can look at the human body and think that there's a hand microbiome, a mouth microbiome, et cetera. There's a soil microbiome, a chair microbiome. And so these are some of the things that we do in the lab. And so today we're going to take you on a journey. We're going to tell you a number of different stories about people who study these microbes and microbiomes. And I hope you're going to be really excited to learn more about these, and at the end, really want to learn more and come to our exhibit at the museum, which I'll touch on. All right, so I want to begin my story. I'll back up and say, as Will mentioned, I have this really cool joint position at a museum in downtown Raleigh, where I have a lab that has all glass walls, and so museum visitors can come up into this museum and watch me do science every day. And I also have a position at this university, North Carolina Central, where I can engage students and other faculty members in my research. And so it's a really awesome position. I encourage you to come see me either place. I'm also involved in the Triangle Center for Evolutionary Medicine. You can ask me about that later. And I'm at NC State and Duke University sometimes. So my story actually begins in the deep backwoods of Wisconsin. I grew up in Michigan, another reason to use my hand. But I spent all this time in Wisconsin, and I spent all my time outside with animals, living in the dirt and playing with microbes, unbeknownst to me. But I was actually trying to train the chipmunks and the squirrels and the chickadees how to eat out of my hand. And I quickly learned which animals could be more trainable. And so I started learning about the variation and got interested in behavior because of that. And it turns out that I developed this fascination with lemurs. So anybody who knows lemurs, there are a lot of different lemurs on this slide. And so not only is there variation in all parts of nature, but all these different primates. And so this is how my talk title comes about. So do you smell like a gorilla? How many think they smell like a gorilla? All right, a few, awesome. I've had other people tell me that before. So what I do now is not only get interested in primates, non-human and human, but I'm really interested in the microbes that live on you, because primates use scent to communicate. So if you guys chose a spouse, one of the reasons you were probably attracted to them is because they smelled good. Well, they're not the ones that smell. The microbes that live on their skin drink their sweat and produce these compounds that are volatile chemicals that produce odors. So your microbes are the ones that produce your body odor, it's not you. So the microbes on your skin are really relevant to primate scent and communication, and that's where we're going to be going today. So if we now take one of these swabs and go to the North Carolina Zoo or to Puerto Rico, where we have a field station to study some macaques, we can actually take that swab, touch it to the armpit of some of these primates, and collect all the microbes that live on the skin of their armpit. How many of you guys have armpits? Awesome. I just got 300 volunteers for my next study. I love it. All right, so we can actually take these swabs, collect the microbes on the skin of the primates, and now look and see what grows in a lab setting. And you can see from these plates that humans actually look kind of boring. We have a lot of little tiny white colonies here, mostly a type of Staphylococcus. And then the other primates have a lot more variety in what lives on their skin. And so that's what I was interested in looking at. And I said I didn't just go to the lab and study these on a petri dish. We actually extracted the DNA from all the microbes on the skin of the primates and then looked at the sequence of those to tell us what we had. And what you find is that if you look at humans, we're actually kind of boring. We don't have a lot of diversity. A lot of what lives on us, on our skin, is Staphylococcus, Corynebacterium. There's a little bit of Prevotella and Anaerococcus, but it's predominantly these 4 different groups that live on human skin. We don't have a lot of variation. And in the microbiome world, that's actually a bad thing generally. Variation is very good. And so when you look at the other primates, so things like old world monkeys have a lot more variation. So you see they have a lot more of these black and gray areas because they have a lot of different kinds of microbes living on their skin. And it turns out when you look at those, they have a lot more fecal and soil associated microbes. Well, that makes sense. They live outside and they play in the dirt all the time, right? So when we think of ourselves as playing in the dirt, we think, oh, we need to take a shower and get rid of all that. Well, it turns out primates wound heal faster than humans, so potentially this is beneficial, and I'll touch on that in a minute. Okay? All right, so let's go next. And so one of the things that's different between humans and primates is, like I said, we take showers, but how many of you guys wear product under your armpits? So this is where armpits get really relevant, okay? A lot of you wear deodorant and antiperspirant, and it turns out you're wearing that to either kill the microbes or to produce less sweat, so there's not as much food for the microbes. So less microbes, less body odor, right? So anybody who wears antiperspirant or deodorant, if I take a swab and I swab your armpit when you're wearing it, you probably won't have a lot of microbes that grow on a plate in my lab. If you stop wearing product for a day or two, you're gonna have more microbes growing there. So that wasn't really surprising to us. But what was surprising is that when we used the sequencing technology to figure out which microbes were actually there, it turns out that people who don't wear product have more Corynebacterium than staph on their skin. But people who wear product have the reverse of that. They have more of Staphylococcus, less of the Corynebacterium. But it turns out if you wear antiperspirant, you're probably feeding these chemicals to your microbes and preferentially getting more variation and more different kinds of microbes on your skin. And so you can see more variation here. There's more of these black and gray areas in the people who wear antiperspirant. And these are people who have stopped wearing antiperspirant for multiple days. So that means that it isn't because they have the product on their skin now. It's you're actually selecting for certain microbes on your skin because you used to wear product in the past. So it's really kind of interesting to go forward. And so what does the future hold here?
Ding.
Well, perfect timing. So the odors that I said are attractive to mates are also attractive to things like mosquitoes. And so studying the skin microbes could be relevant for understanding infectious disease like Zika virus, dengue, et cetera. And I said the primates that have a lot more fecal and soil microbes on their skin tend to wound heal faster. This is anecdotal report. I don't have a lot of scientific backing for this yet, but this is one thing that we're studying now. And then lastly, your host genetics plays a role in which microbes live on you, and then those microbes might in turn influence your susceptibility to disease. And so that's one of the things we're following up on. If anybody actually does want to participate in one of my studies, I don't know if you know that there are different types of earwax, both wet and dry earwax. I'm really looking for people, I know this is discriminatory, I'm sorry, I'm really looking for people with dry earwax. So if you have dry earwax, or if you're super excited to participate in one of my studies, come talk to me at the end. I would love some of your samples. And what I want to leave you with is, if you're interested in learning more, we have this really awesome exhibit that's hosted between the Museum of Natural Sciences and NC State University. It's called The Secret World Inside You. It's all about the microbes and how they relate to disease, et cetera. And so please come out and see it at the museum. And if you're super excited about it, we have buy one, get one free coupons, so you can come see us at the table over there. Mary Ann's holding up that. So hope you come see us, and I'm happy to take any questions.
All right.
Hi, thank you. Is it true that you can't always culture every kinds of microbacteria, et cetera? So not everything shows up in those cultures, is that right?
That is true. Certain people are trying to develop new ways to actually get more things to grow in the lab, but just with a general lab practice, it's very hard to grow a lot of different things that are on my hand or in my body. Yes, so that's one of the things you miss when you try to culture organisms. And so that's why the sequencing technologies are so valuable, because now you can actually look at everything potentially that's there on your surface.
All right, one more.
Some up front.
In your first slide, you saw that baboon has only few colonies on the plate. Is
Yes.
it that they don't have enough microbes in their armpit, or?
No, that could just be that maybe what lives on them doesn't grow in the lab that well. These were just really basic standard lab conditions. We didn't try really hard to get what was growing on the plate. That was just sort of to visualize and let citizen scientists and other participants see the microbes. But we actually went through, and that's why we did the sequencing, to really try and capture everything that's there. And they have a lot more variety of microbe on their skin than humans do.
All right. Thank you, Julie.
Thank you.
Now, I would assume it's okay if when people get a beer next, we can also start like an earwax line.
Sure, yeah.
Okay.
It starts over here.
Okay.
Yeah, Mary Ann's got that going on.
Oh, so we'll do it over there. Just get your earwax, and maybe that'll get you an extra beer ticket. No, just kidding. Just
Uh-oh,
kidding.
don't start that.
I'll take that one from you. All right. Next up, we have, and I'm going to do this. I'm going to get it right, and I'm going to do a really good job, I think. Andrea Azacrate Perel, PhD, is an assistant professor of medicine in the GI division at UNC Chapel Hill. She is also the founding and current director of the UNC Microbiome Core Facility, which provides research and technical support to investigators interested in addressing the roles of host-associated microbiota in health and disease. Andrea, you ready? Yeah, help welcome her. There you go.
All right, thank you. Wow, there's a lot of people here. Okay, so when Julie actually contacted me to present here, I thought, oh, this is great. So I can tell not my story, but I'm going to talk a little bit about my passion and a little bit about an idea, because this is right now an idea or a theory, however you want to call it. So it all started with this graph right here. And what you see here is, and this is from a paper from 2002, that if you look on the left, these are all infectious diseases. If you look on the right, those are chronic diseases. So we've had a dramatic decrease on diseases produced by bacteria or caused by bacteria. But we do have a dramatic increase in diseases that are chronic, like multiple sclerosis, Crohn's disease, diabetes, asthma. And this paper is from before we knew that each one of these diseases were actually related to the microbiome. So this is very interesting. So, wow, what's happening here? When I saw that, I'm like, oh my God, what is happening here? So I started thinking. I didn't have to do a lot of thinking, not especially, you know, today everybody talks about the microbiome. If you Google microbiome, you're gonna see a million news daily. We live in this society. And I'm not talking about just junk food. I'm talking about food-like products too. We are all germophobics. We love our antibacterial soap and what is the other thing that we use in your hands? Sanitizers, which I hate and should be banned. And then, of course, we overuse antibiotics. We take a lot of antibiotics. We feed our animals with antibiotics, so really, really bad. So there are a lot of, you know, publications and papers that are demonstrated that there's something that we're doing that is not right. And so this is where my idea comes. So we went from the medieval times, where people used to just throw their waters, if you know what I mean. Yeah, I mean pee, out the window. But we went all the way to the other extreme, where we want everything clean and no germs. So you see, when we eat, actually what happens, and especially when we eat those stinky cheeses and yogurt and sourdough bread and even you know, fresh vegetables, we're not only feeding the host, we're not only feeding us as humans. This is my daughter. But we're only feeding our gut bacteria. So that's my hypothesis today, that our gut bacteria needs sort of a reseeding. We need to feed it. And what happened is that at one point here, we had those evil, food companies and formula companies that told us, oh, formula is the same as breast milk, or processed food is the same as real food. Food fast or easy and clean. Who has time to cook? Everything is natural or made with natural ingredients, right? So what happened? We stopped cooking. We stopped eating stinky cheeses. The yogurt really doesn't have a lot of bacteria anymore in there, and it should be made with bacteria. Well, at least we're drinking beer, and the beer has enough yeast, so that's really good, yeah. So, but this is what happened. What happens is that communication, that seeding that was happening, just is broken. So basically now we are having all of the diseases where we have allergies, we have eczema, we have asthma. And it's because we live in this society that is too clean, and we're not eating the right foods. So even when we're cooking, what happens is that we have the surface of vegetables or whatever it is that we're cooking is covered with bacteria. And those bacteria are feeding our gut microbiota. So it's very important that we keep feeding it. So basically, that's what we need to eat. And that's the title of my talk: How Probiotic Foods Feed a Healthy Gut Microbiota. So thank you. And I know that this is supposed to be like a sales pitch, but I direct the microbiome core. So if somebody's interested in having their microbiome analyzed, just contact me. Thank you.
We're going to just start a lot of volunteer lines afterwards. 2 questions. One right here. All right.
So can you explain a little bit better about how the foods that we're eating are not good for our microbiomes? And if you were to change the diets that we eat, how should we change our diets to feed our microbes better?
That's a really good question. So eat a lot of fermented foods. So yogurt, but real yogurt, cheese, sourdough, kimchi, kombucha, everything that has bacteria in it. But not only that, we need to cook more. We need to prepare food from fresh ingredients. Because when you're cooking, when you're preparing the food at home, those raw ingredients have already bacteria. We're not, and when we cook them, of course, we want to get rid of the potentially pathogens, like, you really need to cook your chicken. But you really need also to eat bacteria. Yeah?
All right, one more. You're the closest walk. So.
Is it known how much bacteria we should put into our systems? I mean, like if you're drinking kombucha, do you need to drink a whole bottle or just a third of it? Or can you overdose on microbiomes?
Well, if you overdose, be assured that your tummy is going to tell you that and you're going to be in the bathroom a lot. So basically, do not worry about that. Just worry about, of course, like I said, you want to wash your hands. I'm not saying, stop washing your hands or stop cooking your chicken or I'm not saying that. So it's just an equilibrium, if you will. So eat your commensal microbiota, eat your probiotics, eat your prebiotics, and feed also your microbiome.
Nice job. So for those who have come quite often, or at least once before, you know that I love to tell a good joke, and I feel like there's like 2 people that clapped. And there's no better time to tell one than right now. I don't have one. Okay, no, what? Yeah, no, I have one, don't worry. Don't worry, I had to remember it. I went blank there for a second. I got joke fright. Why do chicken coops have 2 doors? There we go, there it is. Why do chicken coops have 2 doors? Nobody? You guys are all supposed to be smart. Come on, let's do this one. If they had 4, it'd be a chicken sedan. You also eat Laffy Taffy, don't you? Yep. I knew it. Don't add me on Facebook, because then you'll know all the answers. I post a daily joke every morning. I've lost a ton of followers and friends. All right, Dr. Lawrence David is an assistant professor in the molecular genetics and microbiology at Duke. His lab works to develop tools and theory for controlling microbiome communities. Ladies and gentlemen, help me welcome Dr. Lawrence David. Thank you, sir.
Go back. Too far. All right. My PhD is actually in computer science, so it always bugs me when I can't handle these things. All right. So this is what your microbiome probably looks like over the course of a year. This is a microbiome in someone's gut measured every day for 365 days. And the different waves here, these different colors are different species. It doesn't matter who they are. And you don't really need a PhD to analyze this, right? You can see that, well, whoever this is, it looks like they had a lot of gnarly green bacteria show up one day. That was probably the time you made a bad decision. You ate that, one too many tacos, or there was that time over there, down where all these blue bacteria grow up and you run out of purple bacteria. That was the time when you went to the Caribbean and you just started eating a lot of funny things and you think it did something funny to your stomach and it turns out it actually did. I know this microbiome really well, actually, because you can probably guess it's mine. I collected it when I was a graduate student 7 years ago, and I was younger and very impressionable, and I had a very persuasive boss. And one of the things that always struck me about this, and I show it a lot, and I return to it, is, and maybe this is just narcissism, but I always thought there was something really elegant and beautiful about this. There are all these little waves and peaks. And having trained, I guess, as a computer scientist and an engineer, someone who studies data a lot, I've always wondered, well, what's causing those? Can we control those peaks? Is this the kind of system that we can take a hold of and know what's going on? So that question was something that stuck with me for so long that I decided to try and make a career out of this and figuring it out. And around the time I was doing this experiment, I started getting really interested in diet. It was around when KFC invented the double down. I don't know if you guys remember that. It was 2 chicken patties sandwiching a hamburger patty. And I thought, wow, this must be doing something to your microbiome. Let's do a diet study and figure this out. And this is what we did. It turned out the university wouldn't let us actually use the double down. They thought it was too dangerous. And so instead, we developed these 2 kinds of diets over here. The one on the left, looks pretty wholesome. I think Andrea would approve. It's got grains and legumes. The one on the right is the one that I personally prefer. It's got a lot of barbecue. It did have cheese, so it is fermented, eggs. And what we did was we fed them to people, these really brave volunteers who would eat them for an entire week. And one of the things that really surprised me was how quickly people's microbiomes actually changed when we put them on these diets and they ate them exclusively for a week. It turned out that almost overnight, the compounds that bacteria make that feed our gut cells actually changed. We changed how much they were being made, as well as different kinds of bacteria were present. And it turns out that some bacteria that actually in mice cause inflammation show up within a single day when people were eating a lot of the diet on the right. So that really stuck with me, that we can have these big impacts on microbial communities inside of people by changing diet. Now in my lab at Duke, though, one of the things we think about is instead of having really big effects on communities, can we have really targeted and specific effects? And the way we do that is we use machines like this. So folks in my lab have spent actually the last couple of years developing these kinds of bioreactors as artificial human guts. So bacteria actually live in here. They're these liquid slurries. They turn around. You can guess that thing on the top right, that's a commercial grade snorkel. And you can imagine it's sucking air out of here because we actually had all these complaints from neighboring labs that thing smelled like death. Actually, it doesn't smell like death. It smells like something worse than that. So it's been nicknamed the bioreactor. And we can do amazing things with this bioreactor. We can put in very specific amounts of bacteria. We can take out things. We can add single kinds of fibers. And we can test these really kinds of sensitive potential interactions between what we eat or the drugs we take or the probiotics we use and specific bacteria inside the human gut. Now, what keeps me up at night, though, is that people, of course, aren't as neat and clean as these bioreactors, unfortunately. And in fact, the kinds of experiments that keep me up are specifically ones like this. So this is my favorite, or actually I should say one of my 2 favorite experiments that keeps me up at night. It reminds me what's going to be hard going forward about designing treatments for the microbiome. This is my son, and this is him hanging out in his grandpa's hometown in the Philippines, eating a fried frog, which is delicious. To me, it evokes sort of what's going to be complicated about the microbiome. Everyone's different in terms of age, genetics, travel, diet, bacterial exposure. And one of the emerging theories, and I think things that you'll notice in the coming years, is whether or not we should be targeting our bacterial therapies and our diets for all these kinds of differences that you see inside of people. Now that sounds like a really tall order. How do we do that? We do have some tricks up our sleeve, and one of the things that's really amazed me, and I'm glad Julie talked about this earlier, is that sequencing has turned out to be this incredibly powerful tool for studying microbiomes. This is a MinION sequencer, so these are cutting-edge sequencers that exist nowadays that in real time, so you can actually load a bacterial slurry into these, they plug into a USB drive, so they're not going to work on the new MacBook Pros, and they can tell you within a single day how, what your bacterial communities look like inside of you. So what you can imagine in the future, what will probably be the case, and maybe even in the near future, is you'll take a drug or try a different kind of diet, and the very next day you can see whether or not it affects your microbiome. So, why will that be useful? Or, I return now, and I'll end on this slide where, I envision one day that we can use techniques like that and technologies like that to figure out, well, how do we actually control these different kinds of peaks on these graphs? Many of these bacteria are associated with health and disease. I'll mention that, at least for me, going back to my sort of passion, why I do this in the first place, I've always just thought it's fascinating, the notion that could we control any of these individual bumps and grooves? Because if we can, I would argue that anything you can control, you've understood, a little bit, right? And these bacteria are part of us. And by extension, therefore, we've understood a little bit more about ourselves. So thanks.
All right, I'm going to start a little bit further back. We'll go here, and then if somebody back here has one question.
Thank you. That's really interesting. Have you used your bioreactor to study the effects of chemical food additives like preservatives or emulsifiers on the microbiome?
No, we haven't done that yet. There's a pretty long laundry list of things we're knocking through, but that's an interesting idea.
What are your thoughts on what potentially those effects would be?
Well, so one of the things that we noticed back in this diet paper is that emulsifiers that we naturally make called bile acids that help us break down fat actually tend to be weakly antimicrobial. And so it wouldn't surprise me if they had in fact, I'm pretty sure they have an effect. But it turns out that they are often needed, if you are eating a lot of fat.
All right, one more. See if anybody back here has one before I turn around. Going once, twice. Nope. Oh, right here. See what now? I just know. There you go.
So what do you think about these probiotic pills that are 15 billion, 25 billion? What's up with those?
They're super interesting. So I'll mention that it's a very political response. So one of the things that I'll mention that happened in this study is that I ate yogurt, and I would track when I ate it. And one of the things I observed is what people often see, which is that you can detect the bacteria that are in probiotics, or at least in yogurt, and then they don't seem to stick around. They pass right through. So people have shown that there are effects of probiotic bacteria when they get to the gut and they're still alive. But nevertheless, I think one of the big challenges going forward is figuring out how to get them to actually hang out and live in the gut. I often think of the analogy of buying a dog at the pet store, right? If you buy a puppy at the pet store, you could release it out into the forest. And it'll probably hang out for a little while, but it's probably not going to colonize the forest. And bacteria that are often showing up in probiotics, they might not necessarily be adapted to living in the gut.
All right, Lawrence, thank you so much. Take that from you. All right. Next up, we have Kirk Beebe. He has been an active scientist for over 2 decades with an overarching interest in applying innovative technologies to fundamental biological questions and clinical needs. Please help me welcome Kirk to the stage.
All right. Okay, so first, just real quickly, especially because we're at this nice informal event, I I'd just like to mention that the views that I'm going to express today aren't really my own, but I do think they overlap a lot with my employer. So you're not gonna hear a lot about the microbiome for a little while, because I actually came into this about halfway through my scientific career. The first half was very focused on just these 4 letters, basically these letters that comprise the basis of our genetic code. And really just my focus the first half of my career was really on these letters and how they played out through our genetic code and our DNA into ultimately comprising a phenotype or a trait at the very end, essentially making us who we are. So this was really my focus. And about 6-7 years into my career, there was an initial kind of invasion into this very simple reductionist idea that this code just played out into everything that we are. And these unwelcome invaders were in the form of these other RNA molecules, starting with RNAi and then microRNAs, which maybe some of you have heard of. These are other RNAs that are coded by our own genetic code, but they don't play out in that nice flow of information. So, this was the first kind of shot across the bow to me realizing that it's really not that simple as this clean flow of information from our genetic code to who we are. So I began to embrace this idea and started to embrace the idea that things were more complex. And right about then, I moved to this company just down the road here, Metabolon. And Metabolon actually is a company that looks at this little purple ball over here of metabolites. Basically, things like glucose and cholesterol that we all go and we get our blood drawn and we get tested, but there are actually hundreds of other metabolites that circulate in living systems. And these metabolites actually help you understand the genetic code in the context of health or the phenotypic state. So I really started to embrace this idea that things were more complex than I initially thought. And I started to embrace it with this technology, and I really liked it. And that's when kind of all hell broke loose, at least for me. Because this is a complex situation to begin with. Just understanding what our genes do, right? And how it influences the phenotype. And a couple years into Metabolon, this is when these swarms of bugs started to appear. Not the bugs that are pictured on this slide, but the bugs we're all talking about here today, the microbiome. You started to see a lot of papers come out talking about how it was important in all kinds of different traits or health states. A lot of papers by Jeff Gordon and others talking about how this was influencing a lot of different things. And this started to play out, and as many of you might, and this is maybe why there's so many people here tonight, started to play out in a lot of popular press. You saw this in magazine articles, news articles, all over the place you started to see this. And my reaction in kind of seeing this initially and seeing this kind of play out was it wasn't really, I didn't greet it with a lot of enthusiasm as it just was all over the place. Microbiome, microbiome, microbiome. Again, going back to the fact that I focused the first half of my career on really trying to understand the genetic code. I got quite irritated about seeing this. My response was, in essence, can't we just kind of take a break and understand, understand our endogenous genetic code and the endogenous molecules that make us who we are before we introduce basically orders of magnitudes of complexity in these stupid microbes. I was quite irritated, frankly. But what happened is, I didn't really touch on it, but I work at a company that has a technology, and we work with a lot of different investigators in pharma and biotech and academia. And what occurred is I started to work with a lot of these clients and also review the literature. And there just was no stopping this train. You could argue and quibble with the details of individual papers, but this really was apparent that there was something real here. And that finally got me to kind of stepping back and not being stubborn and saying, okay, well, let's think about this a little bit more. And I began to kind of, and Julie touched on kind of how this, how sometimes people like myself have to dive into thought here with her primate interests, but I really started to think about this problem. And what occurred to me as I stepped back is that I was actually working with the technology and kind of suddenly immersed in this space. And I was actually in a fortuitous spot. Because if you really think about how microbes work in all kinds of communities out in nature, they often utilize small molecules, basically metabolites, as a currency or a weapon or a language. And this is obviously, as it's in a bit of an ad hoc way, it's played out when it comes to human health and disease as well. There are many, many examples where metabolites, let's say something like, Lawrence, you mentioned bile acids or glucose and things like that are affecting human health. It's actually, Andrea touched on it. It touches on the things we ingest, we eat, whether it be something in red meat or a drug or artificial sweeteners, all the way to molecules that these microbes make to fight off invaders or help us shape our immune system. So just one quick example I wanted to just cite, because I think it's something we can all relate to, that we were able to participate in. It came out just last week. And this was in the area of the concept that a lot of us are familiar with, which is yo-yo dieting. I think our MC here, based on his picture at the beginning, might be dealing with this a little bit. All right, just all right. You brought it up. So, but you know, the whole concept of we get large, we go on a diet, the next time around we gain it faster and we gain more. Why is that? Well, this was a mouse model study, which in essence suggested that part of the challenge is there's a really persistent change in the microbiome. And accompanying that persistent change were changes in small molecule metabolites that help us burn calories. So this is the nice connection that we've seen in using the technology and working in this space.
Ding, 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 ding.
I will leave on this last point. Of course, we all talk about this, how there's a lot of hype in this space. Is it real? Is it not? Frankly, I think it's really just about where we're at with the science. I think at times science can be like cultivating fine vintages where we understand the molecular mechanism and we can act on that molecular mechanism to impact human health and disease. Right now, the microbiome space is about clearing the swamps. It really is a messy time where we're there's a lot of cartography, trying to map it out, figure out where the important areas are. But I think those of us who've been able to look at the evidence and consider it and think about it a lot, I think that we are on fertile soil. It might take a long time to get there, but the reality is I think there is fertile soil in that space. And you know, I guess for me, I'm going to keep wielding this molecular machete of metabolomics, trying to understand it a little bit better. And that's it.
All right, 2 questions. 2 questions. Oh, there you go. All right.
Of our metabolites, the ones that we eat, we naturally produce, and like the microbiota produce, what's the relative breakdown there? Are there many that only come from our microbiome, or are most produced by us or consumed by us and just used by the microbiome?
I think the reality is we don't know for sure, but just in terms of like what we profile, it's the vast majority of metabolites in our body are endogenously produced and or dietary compounds that we bring in. But there's a good proportion that are acted on by the microbiome. If I had to throw out a number, I'd say a good 10, 15, 20% are acted on or influenced or directly produced.
And one more. It's okay, it didn't hurt my feelings. We can stop. I can hear like a pin drop. Why did I gain weight? No, I'm just kidding.
How close are we to cultivating a healthy microbiome with small molecules versus an approach like a fecal transplant or something where you import a microbiome into the host?
I'm not on the front line of that, but I would be shocked if we were close. That's the bottom line. I think we're still in the swamps here.
All right, Kirk, thank you.
Yep.
Thank you. All right, last but certainly not least, Nathan Cude is a microbiologist and team leader in the Microbiome Discovery Group in the BioAg division at Novozymes North America. He leads a team that targets the isolation of microorganisms for agricultural applications using classical and novel isolation approaches and high throughput genome sequencing. Sounds a lot like my bio, to be honest with you. I feel like you kind of copied this one off of the website. All right, Nathan, you ready? Last but certainly not least.
All right, that's a tough act to follow. But so a lot of what we've talked about tonight has been about the human microbiome. But really everything around us has a microbiome, especially soils. And so what I'm going to talk about tonight is how we're using soils to try to feed the whole world. That sounds pretty ambitious, doesn't it? But that's what we're going to talk about. So back about 1510, Leonardo da Vinci said, "We know more about the movement of celestial bodies than about the soil underfoot." And I think that's certainly true. When we think about the advances that we've seen in next generation sequencing, the complexity that we find from just the soil underneath us has just really baffled everybody. I mean, it's been sort of something that we've ignored for a long time, and as we sort of continue to investigate it and dig deeper, the complexity is humongous. For example, in a tablespoon of soil, there's about 50 billion microorganisms. And I mean, that's an astronomical number. Of those 50 billion microorganisms, there's about 30,000 different species. And to sort of put that into perspective, there's about 5,500 mammal species on the entire planet. So a tablespoon of soil represents a humongous genetic diversity with lots of different applications and lots of different functions. So really, even just a small amount, we have, like they were saying, we're in the swamps. We have no idea what's going on inside, even just a small amount of soil. But by using these next generation sequencing techniques, we're starting to get an idea of who's there and maybe beginning to look at what are they doing. And one thing that we know that they're doing is that they're colonizing plants. So we spoke a lot about human microbiomes, but just like we have a microbiome in our gut that help us digest food, plants have a microbiome that colonize their roots. And these plants actually stimulate and change how plants grow. And they do this in three main ways. The first is by making nutrients more available. So they're actually changing molecules in the soil environment and making them more available for the plant to take up and utilize. They actually will fertilize the plant, producing plant hormones to modulate how the plant reacts to its environment and how it grows. And they protect the plant from other microorganisms. So there's we know a lot about how microorganisms can cause disease, but many of the microorganisms in the soil will actually colonize the plant and protect it from invaders or other disease-causing agents. So this has huge applications in agriculture. There's a lot of different challenges that we're coming up to in agriculture as a field that we think can be sort of attacked using the microbiome. So we can look at how can we use these organisms, this diversity in the soils, to try to increase the yields from agriculture. Because that's what a farmer's end motivation is they want to get more yield from the same amount of land with less inputs. So how can we do this in a sustainable fashion? There we go. So there's 4 key challenges that are affecting global agricultural production as we move forward into the 21st century. The first is a growing population. By 2050, the world population is estimated to increase to over 9.6 billion people, and we have to find a way to feed all of these people. Next, and we all know that the climate is changing, and this is going to make crop yields less predictable over time. So as the world heats up, the available land will also change. So we have a decline in total arable land by about 2/3 by 2050. This is also partly due to the increased population, where you have increased urbanization taking over lands that were formerly farmland. And finally, on top of all of that, we have changing economies and changing diets that are leading to more meat consumption. And meat consumption in and of itself requires more field inputs than just grains by themselves. So together, we're looking at a lot of different problems within the agricultural sector, which is going to result in us needing to double the amount of food production in the next, well, probably 50 years. So it's not all doom and gloom, though, because we think we can use the soil microbiome to try to increase the yields that we're producing in the current lands that we have with less inputs overall, and by doing this in a sustainable fashion. So there we are using our microbiome. So now into sort of some of the things that I do. So we take soil samples like that we get from all across the United States, and we isolate lots of different microorganisms from them. We have lots of different strategies for doing this. I don't know if you guys saw, we had a piece where the local PBS station came to to Novozymes to do an interview, and I was there for just a second, and they cut down everything I said to, look at the pretty colors. So I was on there for about 3 seconds, but look at the pretty colors. So there's a huge diversity of microorganisms that are in the soils, and they're all doing lots of different things. But that diversity is so huge, so it means that we need to do huge screening projects in order to look at how all of these different organisms are able to affect plants in different ways. And so we do that in a lot of different ways. The first is by studying these things directly on the plant. So we have here 2 different plants that we're testing, both of which have been introduced to a pathogen. But of course, the one on the right we'd like to highlight was also inoculated with our microbe. And as you can see, that it's prevented that organism or the pathogen from killing the plant. In addition to just pathogen stresses, we also have organisms that are looking at nutrient stresses, drought stresses. So there are organisms already available, natural organisms in the soil microbiomes that can be utilized to increase plant yields from lots of different stresses and help us address a lot of the changes that are going on in global agricultural needs. But what you do in the greenhouse doesn't always work in the field. Like, if I can do this at a very controlled experiment in a greenhouse, can a farmer do it as well? So we need to really scale up the way that we're screening these sort of microbes. And the way that we've done that is by taking the organisms that we're isolating and actually going to the field. We do hundreds of thousands of field trials all across the United States every year to really look at thousands of different microbes in different environments to look at how they can affect how a plant can grow and whether or not they can increase the overall yield of that plant. And all of this is to put together to try to help a farmer get more out of his field with less inputs in order to address what we think is going to be a growing problem over the next century. And a lot of this plays into our overall corporate strategy or purpose, if I can get to it, which is to find biological answers for better lives in a growing world. So it is possible for corporations to address some of the bigger questions, some of the bigger issues that are affecting our country or in our world in a sustainable fashion. So thank you.
I didn't have the heart to do it. Like, it was 4:59, and then whoa, pre-celebration. It was 4:59, and then you said, well, I'm going to tell you a little bit about what I do. And you went for another couple of minutes. I didn't have the heart to do it right at the 5-minute mark when you said, I'm going to tell you what I do. I wanted to give that to you. All right, I see one. Oh, man. Are all y'all going to hang out afterwards a little bit so they can ask questions? I feel like there's been tons of questions I've not been able to get to, so they will hang out for a little bit if you have questions.
I read on the internet yesterday that we have a serious, serious reduction in our biodiversity, both in plants and animals. How is that going to affect what you're doing?
Well, I think a main focus of what we're doing is to try to introduce biodiversity back to agricultural areas. So we're taking biodiversity that's naturally present in certain areas that are beneficial to those plants, and then introducing them to new areas so they can be beneficial.
Well, doesn't that mean, though, that plants and animals are dying? A lot of animals, wild animals, are on the verge of extinction, and that's happened with plants as well. So if they're gone, how can you replace?
I'm not sure if, are you saying the plants are extinct or the microbes
Yes.
are extinct?
Well, the plants are.
Yes, absolutely. I'm not sure, what's your question?
So how are you going to improve crop growth if there's fewer plants growing? We have fewer variety of plants and animals to deal with.
I think oftentimes the ways that the different microorganisms are able to colonize the plants and influence their growth can be shared between very commonly related organisms. So plants that, different types of tomatoes can also benefit from similar microorganisms. So our overall strategy would be to try to find things that can give a broader yield increase, to increase overall food production.
So with whatever's left, it'll be better, essentially, right?
I'm all for conservation.
I told you there'd be one curveball. One curveball question. All right, yes, ma'am.
Hi, I was wondering what is known about what effect the different microbes from different climes have on, say, a given vegetable. So say you have a cucumber that's grown here versus a cucumber grown in Argentina. Is the soil microbe community going to actually impart different nutritional aspects to that? And do we know much about that? And is it good to eat things that are closer to you because that's what your body is used to?
I think that's a great question. What is known is that oftentimes when you'll sequence samples from different locations, the microbiome in those different locations will be different. And even within that, if you isolate specific organisms, if you isolate the same organism from two different locations, they're not genetically identical. So there is a lot of variation between different locations across the world, and the way that they help plants would also be different. So yes, I think there's a lot of variation, and that's why we're looking all across the United States to try to find the next good microbial inoculant for plants. Thanks.
All right, Nathan, thank you so much. Thank you, sir. All right, I won't keep you much longer. Thank you all for hanging in there this evening, and certainly thank you all for coming, and thanks to our speakers for talking this evening. I just have one quick announcement. Next month, it's on the third Thursday, RTP180 will happen again, and it will be virtual reality, which I'm told will be out of this world. I snuck one last one in there. Y'all have a good night. We'll see you next month.